Welcome to ASM Live from the 2014 American Society for Microbiology meeting in Boston, Massachusetts. I'm Stanley Malloy, I'm your host today. We're going to talk about some things that are important, interesting, and some surprising aspects of microbes. Have a fun conversation dealing with these types of problems. I want to encourage everyone in our studio audience to feel comfortable asking questions. Just go over to the mic and we'll get your question when we have a chance. And if you're listening online, please send questions via Twitter to #ASM2014. Today, we're going to talk about pathogens on planes. And our guests are Kiril Vaglenov.
Correct.
And Jim Barbaree, both from Auburn University. And they've been working on issues of pathogens lingering on planes for a long period of time. Kiril, could you give us a brief summary of what you've discovered?
Well, we are not surveying the presence of pathogens on airplanes. We wanted to figure out what's the survivability and the possible transmission of two very important human pathogens like MRSA and E. coli on airplane cabin surfaces under the air cabin conditions. So we figure out that these pathogens can remain on these surfaces over a week, and there is a high risk of them being transmitted from these surfaces to the fingers of the passengers. Of course, we didn't take in account whether or not if we disinfect those surfaces, what would happen. So it's just a baseline for establishing a risk assessment, and our future plans are to check those protocols.
Let me also add that this comes from a desire to look at the risk. What is the risk for the passenger? The person who's on the airplane is the one who is concerned about being in a crowded space and what is my risk. And so this project was a database type of gathering project where we looked at survivability and also the transmissibility of two different pathogenic bacteria.
So Jim, why would survival on an airplane environment be different than survival, on the table here or on our couches or other surfaces?
Well, it may be a little different according to the environmental conditions. Environmental conditions for bacteria on an airplane cabin are not that good in that the humidity is low, around 20% usually, and the temperature is room temperature. And some bacteria require higher humidity, and so we were interested in these conditions, how long these bacteria can stay on these surfaces. So we got the surfaces from the airlines.
So one of the things that is often done when people want to make guesses about survivability of pathogens is they'll use a non-pathogenic strain as an indicator of the survival. You used the bona fide pathogens, two reasonably nasty pathogens. Why did you do that instead of using their less virulent cousins?
Well, that would have been a possibility, and of course we are planning to do non-pathogenic cousins, for example, for Mycobacterium tuberculosis in the future. But taking the precautions which I took, we took our shot and we work with the pathogenic strains because it is believed that some of them, besides their high virulence, are also well equipped with traits that allow them to sustain longer in the environment. Like, for example, MRSA. There have been other studies that have shown even over a year persistence on abiotic surfaces, especially hospital environment, which is slightly higher humidity. So that's why we decided to get as close as possible to the real situation, because after all we are doing a lab study. So.
Well, you always have questions. if you do not use the organisms themselves. There's always a question, is this the same response that I would get from another bacterium? And so we had the opportunity to do it with the real pathogens. And these pathogens do exist, and there are outbreaks of E. coli 0157, and MRSA is a pretty universal organism itself. So we use those organisms.
Are there examples of outbreaks that are associated with airplanes of these two organisms?
There are not very many examples, none with these two organisms. There is a report of a Mycobacterium tuberculosis transmission. Recently there's some news about MERS virus, which is a virus, not a bacterium. It's very difficult to do any kind of epidemiologic study with airplanes because people get on the airplane, they go where they're going, and then they're gone. And the other thing that's unique about the airplane is that you may have a long flight. You may sit in a room that's crowded for an hour or two or all morning, but if you have a flight to Australia or something like that, you're there sharing microorganisms for a long time.
Now, in the process of doing this study, you chose a few different conditions to look at the survivability.
Yeah.
Why did you choose those conditions, and what were they?
First of all, we tried the surfaces that were most commonly in contact with this human traffic in the cabin, like the tray table, armrest, also the window shade, the toilet handle, which basically allows you to flush, and then we also used a phosphate buffer as a control, also to emulate the possible body fluids presence with these pathogens. We used 2 recipes of simulated sweat and simulated saliva. So we wanted to see the impact of different physicochemical properties of the surfaces, as well as the organic presence of body fluids, such as saliva and sweat, which might affect survivability different because of the pH of both.
It's interesting that, just a side story, just before we sat down to talk, I got a tweet from somebody saying the person next to him on a plane changed a poopy diaper on the tray table, and apparently that's not uncommon. And so you could imagine where these types of things could be very serious.
There are recent studies of E. coli 157H7 outbreaks in daycare, precisely related to people changing diapers and being asymptomatic carriers. So we thought that this is also a possible scenario on an airplane.
Now, when you look at survivability, you can have a few organisms survive, but sometimes it takes a substantial number to cause disease. What is the relative decrease over time that you saw for these two types of organisms in the different environments on a plane?
Well, first of all, let me say that we had to decide on what would be the right amount to put on the surfaces. And we had noticed that some studies used a huge number. I mean, we felt like that was too large, so we used 10 to the 5th power per mil suspensions in our studies, which if we had used higher amounts, we would have gotten longer survival. And so we searched the literature and we made a decision based on that, okay, in terms of the amount.
Before we go on, I want to encourage anyone in our audience to feel comfortable. Please come ask questions at the microphone. And those of you who are listening online, please send any questions to #ASM2014. We have a question from the audience right now.
This is a fascinating study. I'm Michael Schmidt, and the one question that I have is, how often are these surfaces routinely cleaned by our friends at the airlines?
Unfortunately, I can't give you an answer because I don't know. And we haven't really tested any of their protocols for cleaning.
Do we know if, I know the FAA is responsible for health and safety of the traveling public, does the FAA have the equivalent of the Joint Commission that we have in hospitals that require us to clean hospital rooms in between patient stays? Is there such a requirement for planes? And I know you may not know the answer to that, but we'll rhetorically throw it out to our friends at the FDA and we'll wait for their answer.
Well, let me say that part of our funding came from FAA. And so FAA and also the airline industry is very interested in the safety of their passengers. They're very open-minded about any procedures that might improve. So they were very cooperative, and the funding came indirectly from the airlines and from FAA.
That's fantastic news. So the FAA is indeed concerned about our health and safety.
I don't want to say concerned. I would say they want to know.
They want to know.
The question is, what is the risk?
Yeah.
And that's what they want to know.
Well, thank you.
So it is an interesting issue to think about because there's so many potential variables on an airplane that you have to deal with. So for example, if I understand correctly, you found lower survivability in the more permeable surface of seats than you saw on hard surfaces. Is that correct?
Yeah. It has to do with the fact that a lot of times bacteria would try to attach into pores where they can find protection from environmental stress and dehydration, UV light, and in turn, when we did our transmission assay, because these surfaces were porous, the contact with the pigskin proxy model that we used wasn't enough. Basically, these surfaces that were more porous, more irregular, weren't giving such high transfer rates.
Let's come back to this, but first, since you brought it up, you're going to have to explain why pigs are a good surrogate for people.
Well, if we go back to the beginning, you asked me how on earth we decided to use such pathogenic strains. Thought to my advisor in the beginning, well, why don't we just draw an undergraduate student to do that? Of course, this was a joke.
I didn't approve of that.
Yeah, we needed to go through a lot of approvals and protocols for that. Of course, there are studies, people are using volunteers with the right protocol. So I didn't have the time and nerve to go through all this administrative paperwork. So I saw that there were several studies that people were actually using pigskin. And they would sterilize that and use it as a proxy as a contact model for transmission from a contaminated surface to the skin.
Yeah, we're very kin to pigs in that we have some people have heart valves.
Right. Often when you say pigskin, the general public thinks football. So
yeah.
Oh yeah.
And so back to the porous surfaces, I think that's really good news in many ways because the ability to decontaminate those porous surfaces is much more difficult typically than a hard surface.
Yeah,
exactly. Yes, you can sort of divide the surfaces into porous and non-porous surfaces. And the porous surfaces will protect the bacteria more.
Oh, that's great. We have a question from our online audience.
Yeah, this actually comes from the chat room, and I'm not quite sure if it's a question more than a comment. It comes from Gary Taranzos, and he says, I would think that in any place we need to think about the infectious dose, and thus enteric and respiratory viral pathogens may be more likely to be dispersed in airplanes rather than bacteria.
Well, the infectious dose for E. coli 015787 is only 100 cells or less. So it has a very low infectious dose as far as that organism is concerned. In terms of MRSA, you can get different kinds of infections from MRSA, so a skin infection or respiratory, all kinds of infections. So I don't know what you can't really tell what the infectious dose is of MRSA, but we did choose an organism that had a low infectious dose. And I am well aware of the fact that some of the viruses have a low infectious dose. But we did a bacterial study. We're taking one thing at a time. And so we did a bacterial study this time.
We have another question from our studio audience.
Yeah, thanks. Ed Dudley from Penn State University. Actually, this is a really fascinating topic for me because not only as a microbiologist, but I'm also a student pilot. Which, of course, I've been assured by my flight instructor that the little single-engine Tomahawk that we fly probably shouldn't have these issues. But anyways, I told him I was coming to this, and he actually sent me an interesting article that came, I think it was from the National Center for Public Policy Research. And I didn't know, kind of if you had some thoughts about this and whether your study's been looking at some various environmental factors. And what the article was suggesting was back in the days when smoking was allowed on airlines, that the air was actually better back then because we had, it was something like 4 times more fresh air would be introduced into the airplanes than we do today. So I was wondering, if that, some basically changes, such as that the airline industry might be doing might also be contributing to the spread or even maybe even persistence of some infectious diseases.
Yeah, we need to be clear that we're talking about transmission with what we call fomites here. We're not talking about airborne transmission. In the meetings I've been in with representatives from airlines, they tell me they have a HEPA filter system in the big commercial airlines, and that it's very efficient and does filter out the air very efficiently. And their air return is set to be optimum for what they need. So we did not look at airborne transmission, but I did receive that information from the airlines.
Having sat on airplanes in those days when the distinction between smoking and non-smoking was one row, I can guarantee the air wasn't better.
Well, I think we need coughers and non-coughers.
Right.
Well, if you knew my flight instructor, you'd know he could be full of it. So, thank you.
Thank you. We have another question from the audience.
So, again, we're not looking at airborne transmission. So the question that I think, I always get asked is, what do you do to fly safe? So knowing what you two guys know now about the dangers on planes, what do you do to protect yourself as you're sitting in the seat and flushing the toilet and drinking the drink and eating the pretzels or peanuts?
Well, I think the only thing you can do is you can use good hygiene. And I think you can follow your mother's advice and do that. And you're using the word dangers, which is a little bit too extreme, I think.
So that's good news. So on a scale of 1 to 10, how big of a risk do you think this is?
We really don't know. This is baseline data, and this is very difficult to obtain. Like I said before, you have a cargo of people, and then once it lands and they're gone, you can't ask them questions, you can't find out if they got sick later, etc. So if you did a study, you would have to really put a lot of money into it and do an epidemiologic study over a period of time, which would be quite expensive and kind of hard to do because of the situation of logistics of everything.
Once upon a time, there was something called shoe leather epidemiology. So you're now suggesting we got to do airplane epidemiology.
Well, I'm not suggesting we do it. I'm just telling you what it would take in order to do this, and it's very difficult. We all realize that.
Thank you.
It's also true that if you say, what's the risk on a scale of 1 to 10, you have to define what 10 is. Now, if 10 is snakes on a plane, it might be a little different answer.
We've all seen the movie Snakes on a Plane, even on a plane.
Yeah.
No, this type of conditions or communal settings are not met exclusively in the airplane. You might have them at the movie theater, you might have them at the metro, airport, and there are a lot of factors that we have to take in account. Asymptomatic carriers, the time spent together, and the immune state of some passengers, I mean, elderly, kids, and so forth.
Kiril, I think that that's an important point. If you say that survival on an airplane is going to be less because of the humidity, then that does kind of set a baseline to say, if it's surviving this long on an airplane, we have to be worried about it in all of these other potential surfaces where large groups of social
You know,
gatherings
the air humidity is low, but we have to remember that certain microorganisms, certain viruses, certain bacteria tolerate very well the low air humidity.
Right.
For instance, bacillus, spore-forming bacteria.
The other thing is we would like to do some further studies, and maybe we can look at incorporating some antibacterial compounds in the composites for the tray table, looking at the pockets. What can we do to, we already know that there are some compounds can be used in fabrics. So if there's an interest in addressing this situation, then that can be done to minimize it. You never be able to totally do away with this problem.
Even simpler than that, you could imagine the turnaround time for airplanes now is so short.
Yes.
People are off, people are on. You could imagine that if this is a serious issue, that you could add wipe down of the hard surfaces, and that alone could potentially really decrease potential interactions with the key fomites, right?
That would, you might even allow the passengers to wipe it down.
You get on the plane and you get your disinfectant wipe before you go to your seat, right?
And put the passengers to work.
Right. Well, that's a good idea. You could even charge the passengers for the
I
disinfectant.
know. Let's don't do that. It costs enough already.
Another possibility that I was expecting you might say something about is using a physical approach like UV to sterilize large surfaces on something like an airplane.
Well, you can use UV light. We do have some bacteria that have repair mechanisms for UV light, and it's used routinely in biosafety cabinets, places like that. But I don't think it's very feasible to do that.
So what's next? So you've looked pretty extensively at MRSA and at E. coli 0157H7. And what other potential pathogens will you be exploring?
Yes. Dr. Barbaree.
I have a graduate student who has begun work with another species of Mycobacterium. In this case, we are using a simulant because we don't want to use Mycobacterium tuberculosis, and it is too infectious, and it takes a level 3 lab to work with that organism. So we're using another species, a non-pathogenic species of Mycobacterium to do the same things that we've already done. That's number one. Number two, we want to look at disinfection procedures, what is feasible. Can we, we would like to get funding to look at how can we put these antibacterial compounds into some of the surfaces and try to minimize the existence of the organisms on the airplane.
Now with Mycobacterium, that is an organism that's transmitted via aerosol. And so will you be using a different approach to examine its survival in this particular environment?
No, we will use the same approach. We'll do exactly what we've done before. We're not going to do airborne studies.
There have been others who have spent some time sneaking into bathrooms of airplanes and other things and sampling organisms and publishing on their results. Can you take some of those types of studies and correlate them with the things that you've done?
Well, Kiril, you might answer that, your literature search.
Definitely. Certain studies from public laboratories, there have been studies that have looked in factors like splashing and aerosolation and so forth. So we are planning to do something like that in the future. But mainly it's going to be looking at the protocols that the airlines have also, and check how they relate to our data. So that's our plan, because I haven't even started looking in that, checking their protocols, how good they are, because a lot of people have asked me, and I have no straight answer to that.
Yeah,
yeah.
Excuse me, let me just say that this was a microbiological study. Right. And it was to get a database, and that's what we've done. And if you look at what we've done, we've chosen a gram-negative bacterium, a gram-positive bacterium, and now we're looking at a mycobacterium which is acid-fast and is more resilient because of the mycolic acid that's around the outer part of the cell. And so we're just getting into this, and I think we're laying down basic data.
We're nearing the end, but we have one last question from our studio audience.
Oh, I got the last question. So during your talk, you kind of kept talking about the specific pathogens you want to look at. I'm kind of curious to know that on normal conditions, and what kind of microbial communities that are naturally present on the surfaces in airplanes. So do you think, like, say, for example, like you look at MRSA or E. coli 0157, are they the predominant species in natural microbial communities on the surfaces under that condition? Because my point is, if they are not the major players on the airplanes, what's the concerns to look at those species? And also when you talk about the other species, the pathogens you want to look at, I just feel like it may be helpful to pick up the species that are constantly associated with those surfaces.
I think that's a very good point, and there have been some studies of people finding different types of organisms, air sampling. And for example, we do know that when people load and unload from an airplane, that the bacterial count goes very high at that point, higher than at any point in time. And there have been some studies to see which bacteria. Now let me say that we use 0157H7 because it would be a highly infectious organism, okay? MRSA would be an organism that could be present on the airplane. But your point about looking at what's there is a good point. But again, we're looking at a real good basic study with these bacteria we chose.
And going also to that subject, there have been sequencing studies examining the HEPA filters, getting the information that you just asked about. And MRSA and E. coli are obviously not the predominant organisms there. But we have to remember that, especially MRSA and Staph aureus, they are often found in humans. They're about 30% of the U.S. population is actually an asymptomatic carrier of Staph aureus. And I think in a transient way, about or less than 1% of the U.S. population is carrier of MRSA. So there is a possibility that these pathogens would actually be present in the airplane.
Okay. Thank you.
So just to tie things up, I think one of the key things that you've said is that this study is meant to establish the baseline, and not to scare people about the risk, but to say, how can we identify potential pathogens and figure out better ways to eliminate or at least reduce the risk? And that's clearly something we need to look at with microbial populations in many, many different areas, including daycare centers, as you mentioned earlier. So
Yes.
I want to thank our guests, Jim Barbaree and Kiril Vaglenov, both from Auburn University. Thank you very much for joining us today.
You're welcome.
I'm Stanley Malloy. Thank all of you who have been watching online and all of you in the studio audience.
They have some other.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this.
Anybody who has access to this facility first has to go through an IR scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.